All right, now we're going to take a look at the building blocks that make up macromolecules. These are also known as monomers. Mono means one, mer means unit. So these are the single individual units that, when we put them together, can build a larger molecule. Last time we talked about what the macromolecules were called. I'm now going to cover the monomers, the individual units. So we're going to talk about amino acids, which make up proteins, monosaccharides or sugars, which make up polysaccharides, fatty acids, which make up a variety of lipids, and nucleotides, which make up DNA and RNA. Now I'm going to start with amino acids. Amino acids all have this same basic structure. They have a central carbon. So you can see the main part of the molecule here is a nitrogen, a carbon, and another carbon. So in the middle is our central carbon. It has an amino group, and it has a carboxyl group. Now, if you recall, the carboxyl group is acidic. So right here, we have the origin of the name of this molecule. It is an amino acid. So it's named for its functional groups. Now, this R group can be any number of things. The R group is what gives each amino acid its unique chemistry, its chemical properties. So here are 4 examples. There are actually many more amino acids than this, but you can see that each one of them has a central carbon, an amino group, and a carboxyl group. But what differs is this part that's highlighted in pink. This is the R group. So this is a non-polar R group, which means that this amino acid is non-polar. It's going to be hydrophobic. It's not going to want to hang out near water. Same thing with this one. This is non-polar, and therefore this is going to want to hang out with lipids, not water. These are polar R groups. So this one is polar. This amino acid therefore likes to be around water or other polar molecules. And same thing with this one here. Now, there are other things that are other chemical properties that R groups can give an amino acid. We're not going to go into all of those, but if you ever take a biochemistry class, you will undoubtedly look at these properties. This is the 20 amino acids that we use in our body to make all of our proteins. So if you look at this, the top part of each one of them, again, is the amino acid. Here's the amino group, here's the central carbon, and here's the carboxyl group. All right, so you can see that that's the same on all the 20 amino acids, but the part that's going down from there, the R group, is different for each one of these, and there's quite a lot of variety. That lets us build all kinds of proteins. Okay, let's take a look at monosaccharides. Mono, again, means one. Saccharide means sugar. These are also often called simple sugars because they are single units of sugar. Monosaccharides are always take the shape of a carbon ring. So remember, the carbons are the bend in the molecule here. A carbon ring that has one oxygen in it, sort of like a necklace where this is the jewel hanging down. The other thing about monosaccharides is that they have tons of hydroxyl groups. So I've circled all of the hydroxyl groups in this particular monosaccharide, which is glucose. And that means all of these hydroxyl groups, because they are hydrophilic, that means that sugars dissolve easily in water because they're covered with polar functional groups. Oh, sorry. One other thing on that. The last thing that can help you identify a monosaccharide is that they always have a 1:1 ratio of carbon to hydrogen to oxygen. So this one in glucose there are 6 carbons, right? 1, 2, 3, 4, 5, 6. There are 12 hydrogens. 1, 2, 3, 4, 5, 6, 7, and the other hydrogens are not drawn here because we tend to ignore them. It's hydrogen discrimination. So this would be 8, 9, 10, and 11. And where did I lose my 12? Or did I think I messed up my counting. Anyway, there are 12. And then there are 6 oxygens. All right, so if there's 1 carbon, there's going to be 2 hydrogens and 1 oxygen. If you double that, it's going to be 2, 4, 2, 3, 6, 3, and so on. Most people don't bother counting. The ring of carbon with the oxygen and the hydroxyl groups is usually enough to clue you into the fact that it's a monosaccharide. Okay. Now, there are a number of different monosaccharides. They vary in terms of the number of carbons in the ring. So this one has 5 carbons in the ring and 1 dangling off to the side. This has 4 carbons in the ring and 1 dangling off to the side. Same thing with these. And the other thing that distinguishes them is which direction the hydroxyl groups go in. So glucose has this one down, up, down, down, but galactose, which otherwise looks very similar, has up, up, down, up, and that makes it a different monosaccharide. You do not have to know these structures, but take a look and see if you can see what they all have in common. They all have this carbon ring, lots of hydroxyl groups, and the carbon ring is closed with an oxygen. All right. These 2 sugars, by the way, are not typically part of our diet, but these are what we form RNA with. The R in RNA is ribose, or DNA. The D in DNA is deoxyribose. So these sugars are going to come back into the story later on. Okay, let's take a look at fatty acids. Fatty acids are long carbon chains. They can vary in length. Some of them are like 4 carbons long, some are 24 carbons long. So the length varies, but they all have these zigzag things. And at one end, there will be a carboxyl group. That carboxyl group is how this fatty acid is going to attach to other molecules to make something bigger. At the other end from the carboxyl group is a methyl group. Now, remember, we're kind of ignoring the hydrogens because we discriminate against them. They're invisible. So if there's nothing else drawn here, the assumption is that this last carbon is bound to 3 hydrogen atoms. Okay, fatty acids have different carbon chains. Some of them, every carbon has 2 hydrogens, so every carbon has 4 bonds total. We say that this is a saturated fatty acid because every carbon is saturated or has as much hydrogen as it can hold. Sometimes a carbon will form a double bond with its neighbor, and the hydrogens that would have been part of that bond are gone. So these are considered unsaturated fatty acids. Sometimes that double bond still allows the molecule to be straight, and sometimes that double bond causes the molecule to bend. When it allows it to be straight, we call it a trans fatty acid. Trans means across from or opposite. So this hydrogen is opposite this one. This is a cis unsaturated fatty acid because both of the hydrogens are on the same side of the molecule. Cis means same side. Now, an interesting thing about these shapes is that when you have a fat made of saturated fatty acids, then they are able to stack close together because they all have the same basic shape. These fats tend to form solids at room temperature. So things like animal fat or coconut oil, which form solids. Those also tend to be problematic in our bodies. The fatty acids that are bent or have irregular shapes don't stack well together and tend to remain liquid at room temperature. And these fats are better for our body and in some cases essential. All right, finally, nucleotides. Nucleotides are made of 3 different groups. Each nucleotide is made of a sugar molecule, either ribose or deoxyribose. It also has a phosphate group, one of the functional groups. And every nucleotide has what's called a nitrogenous base. I'm going to show you in a minute the different nitrogenous bases, but really all you're training your eye to look for is lots of nitrogens. So if there you see a sugar and the phosphate and then something with lots of nitrogens, you have a nucleotide. Now, the nitrogenous base is what gives each nucleotide its unique properties, kind of like the R group with amino acids. So, this is what adenine looks like, and when it's bound to a sugar and a phosphate, it gives us the nucleotide adenosine. This one is guanine, so you can see the structure is a little different. We also have thymine, cytosine, and uracil. Now, we're going to get to know all of these more later, but the point is that all of them have multiple nitrogens, and therefore they are nitrogenous bases. So if you see that with a sugar and a phosphate, you've got a nucleotide. Now, each of these monomers is used to build a polymer or several different polymers. So the amino acids we looked at will, when we put them together, will form peptides or proteins. Nucleotides, when we put them together, will form nucleic acids like DNA or RNA. The monosaccharides, when we put them together, will form polysaccharides. In animals, we make glycogen. Plants make starch and cellulose. Those are the polysaccharides. Fatty acids get put together to form what is called a triglyceride. This is the form that fat takes when it's stored in our body for energy.